Good morning. My name is Ninon Mavraki from Royal Belgian Institute of Natural Sciences. I will present to you our results regarding the food web structure of the fouling communities as they are formed along the depth gradient at a gravity-based foundation in the Belgian part of the North Sea. The establishment of offshore wind farms provides habitat for sessile organisms that rapidly colonize these artificial hard substrates. Different zonations are formed along the depth gradient, leading us to the main aim of the study, which is to investigate the food web characteristics at the ecological depth zones along a wind turbine. In order to assess this, we used stable isotope analysis. So plotting the results from stable isotope analysis leads to this quite perplexed plot, which shows the isotopic and trophic niches of the 6 different communities under study. But this plot is rather complicated, and in order to ecologically interpret it, we need to focus on the overlaps between the ellipses that are formed by the different communities. So in this table, we see the overlaps that are formed by the communities, both as numbers on the lower part of the table and represented as colors on the upper part of the table. The red color represents high heterogeneity and hence low overlap, whereas the green color represents low heterogeneity and high overlap. And the orange color, of course, is the intermediate level. What is obvious here is that the intertidal zone here is completely different from the rest of the communities, whereas the muscle zone that follows the intertidal along the depth gradient is at an intermediate level different from the rest of the communities. Finally, we see that the deeper we go from the Yasa zone to the soft substrate community, there are more similarities between the trophic habits of the communities, with the soft substrate community being the most similar to the rest. Another measure for quantifying similarities or dissimilarities between the different communities is the trophic position, which actually shows the ranges of the consumers in the different communities. This can be estimated by the delta 15 nitrogen values. With the black arrows, we can see the trophic position ranges along the 6 different communities under study. And what we see is that in the intertidal zone, here, we have the smallest range, whereas in the erosion protection layer, we have the largest range, indicating that mobility might play an important role, since in the intertidal zone we mainly find sessile organisms, whereas in the erosion protection layer we find both sessile and foraging species. Focusing now on the species-specific level, and in particular in 2 of the most common species found in the area, we see that there are dissimilarities between the trophic habits of the same species that are occupying different ecological zones. For example, the brittle star of Ophiothrix fragilis has isotopic signatures that expand more at the delta 13 carbon values, indicating that this particular species is taking advantage of different food sources. On the other hand, the anemone Metridium dianthus has isotopic signatures that vary on the delta 15 nitrogen values, showing that this species is occupying different trophic positions at different ecological zones. To conclude, this study reveals that the heterogeneity of trophic structures increases closer to the sea surface, that the trophic position range increases with mobility, and the species-specific studies reveal the depth-dependent trophic plasticity. Thank you for your attention.